Hello, and the topic for this video is going to be the cytoskeleton associated with the cell. Now the cytoskeleton is going to be the components of the cell that are essentially going to make up the internal skeleton of our animal cell, all right? And with components of the cytoskeleton, we've got three types of components associated with the cytoskeleton. The first one is going to be actin. Your textbook also refers to that as microfilaments. I'm going to call them actin most commonly because actin is the protein that makes up this particular fiber, all right? So actin is going to be the thinnest of all of our cytoskeletal components. And essentially what it is, I'm just going to draw it as a pink rod, okay? Its components essentially are made up of repeating units, and that's what I'm doing as the dots here, of the protein actin, okay? And that actin microfilament is going to be involved in cell movement. So with our cell, I'm going to draw a little generic cell up here, smaller than the cell I had in the last one because I've got some other topics I'm going to talk about. Actin is going to kind of run right along the cell membrane. You'll have some that'll extend into the cytoplasm as well, all right? But what those actin microfilaments do is they push on the cell membrane to extend the cell membrane out, okay? And that's going to be how the actin generates movement within our cell, all right? And I should, with our cell, put our nucleus here so it looks like a proper fried egg, all right? But the actin, that's going to be our thinnest of the three filaments I'm going to talk about. It's involved in cell movement. Actin you will also hear about again when you take anatomy and physiology. Actin is going to be one of the proteins that's involved in muscles and getting muscles to contract to get movement associated with muscles. So you'll hear about actin again. But in other cells, actin alone, without the other protein in the case of muscle cells, because muscle cells need both actin and myosin to contract. The cell just has actin, and it essentially sends out those little projections like this and gets individual cells to move, okay? So actin is going to be our thinnest. That's our microfilament. I'll use a different color here so I can draw it on the same cell. We've got our intermediate filament, and it's named because it's in between on size between actin and our largest filament, which will be microtubules, all right? So the intermediate filament is very fibrous, and it has to do with maintaining cell shape and also in anchoring organelles. So if you take the skin on the back of your hand and you pull up on it, you're going to distort the skin and you're distorting those cells, but if you let go, your skin's going to fall back to its normal shape. It's going to be the intermediate filaments that are going to allow that to happen, all right? And just for the record, if you pull up on your skin and it doesn't go back to normal, that's one sign of dehydration. You don't have enough water in your cells, so it's not allowing that protein to function normally, all right? That's one way you can test for dehydration, although by the time you get to that point, you're pretty gosh darn dehydrated, all right? But the intermediate filaments, I always draw the nucleus kind of centered in the cell. And it's not maintained there just by floating. You've got some intermediate filaments that are going to anchor it. You're going to have some intermediate filaments along the cell membrane. And that's going to, intermediate, that is going to help maintain the cell's overall shape. All right, we'll label our actin in pink here. All right, so the intermediate filaments involved in anchoring, it's kind of the middle size of our 3 filaments. Third one, are going to be our microtubules. Okay, the microtubules are made up of 2 different proteins. The proteins that make up microtubules are alpha tubulin and beta tubulin. All right. And the microtubules are a little bit different as far as their structure goes, because essentially what you have is you have dimers. I'll see if I can draw these in 2 different colors that kind of alternate and generate a tube-like structure. All right, so that's my poor drawing or diagram of tubulin. All right, with the one color being alpha tubulin, the other color being beta tubulin. I've got some better diagrams in the PowerPoint to show that, okay? But essentially what it does is the alpha and beta tubulin come together to form a hollow tube, okay? And the microtubules are going to be found throughout the cell. And those microtubules are going to be essentially the way I think about them are they're kind of like the train tracks within the cell. So when I talked about, in a previous video, I talked about how vesicles can move from the rough endoplasmic reticulum to the Golgi apparatus. I drew them just kind of as floating in space. They aren't actually floating in space. They're following tracks of microtubules, so that if we draw in here our endoplasmic reticulum and our Golgi, there's going to be a track of microtubules that are going to connect those so that the vesicle doesn't get lost in the cytoplasm. All right, so they kind of provide a scaffolding for vessels to move within the cell. Microtubules also are going to compose a number of structures within the cell. Microtubules are going to be part of what composes flagella, cilia, and centrioles. And we haven't talked really about centrioles or cilia yet. I talked a little bit about flagella when I was talking about components of the cell way back in a number of videos. But flagella and cilia are going to be involved in movement. The centrioles are going to become important when we talk about a process within the cell, mitosis. So cells being able to divide. All right. And I also think that it's worth mentioning here, the centrioles are going to be found in animal cells, but centrioles are not actually found within plant cells. So that's another difference between plant and animal cells. All right. So I'll kind of talk a little bit about flagella and cilia and the centrioles here. And essentially what you've got when you make up those structures is you've kind of got doublets. All right, and I'm going to kind of draw them this way. Not the best drawing, don't make fun of me. All right, what you're going to have, we've got 3, 4, 5, 6, 7, 8, 9. All right, so we've got 9 doublets, and then we're going to have 2 in the center. So the arrangement for the flagella and the cilia is a 9 plus 2 arrangement of microtubules. And what I've done here is what you're looking at is kind of a cross section. So the main difference that we're talking about between flagella and cilia, if I erase this or smear it around, flagella are going to be longer. Cilia, relatively speaking, are going to be shorter. Okay? So if we have a cell with a flagella coming off of it, and that one cell that we've got in the human body that has a flagella coming off of it is going to be a sperm cell. Okay, that's the only animal cell that's going to have a flagella. All right. Whereas cilia are going to be kind of just short projections off of, say, one end of a cell. All right. And you're going to find ciliated cells lining things like the respiratory tracts. So the trachea has ciliated cells because you want to prevent the dust and other crap that you breathe in from going down into your lungs. All right. The cilia, you can kind of think of them as fingers. They kind of beat and they kind of move in the case of your trachea to keep particles from going down into the lungs. All right. So cilia are going to be shorter, sperm are going to be longer. The part that you're looking at here, if I cut through our flagella like that and we look at it looking at the end, looking at it end on, that's what you'd see. You'd see a 9 plus 2 arrangement there. And what you're also going to have associated with this, and I'm going to just draw this as pink and just as a line because it's easier. There's going to be another protein associated with that flagella. That other protein is known as Dinan. And that Dinan is essentially going to be what allows those microtubules. So if the 2 markers here represent, this is one microtubule, this is one microtubule here, there would be Dinan in between them that kind of allow it to slide next to one another. And it's going to be that sliding motion that allows the flagella and the cilia to move. Okay? So in terms of that, the flagella are going to move kind of by bending. And that bending is because Dinan kind of latches on to the microtubules and shimmies up and down to move the microtubules relative to one another. Okay? The cilia, again, going to be similar in the 9 plus 2 arrangement, but these are going to be kind of short movements. They aren't going to have quite the action that a flagella would as far as movement. All right. Both flagella and cilia are made of microtubules. Where they're found in the body is different. Like I said before, only cell in the human body that has a flagella is going to be the sperm cell, all right, and that's so the sperm can move to meet the egg. Cilia or ciliated cells found a lot in the lung and also the gut, all right, because those are areas that you're either going to want to move, prevent things from getting down into in the case of the respiratory system, or in the case of the digestive system, you want to move those food particles through, okay? Centrioles also are going to be made up of microtubules, but the centrioles have a triplet arrangement. Whereas up here, we had a doublet because we had 2 sets of microtubules linked together. The centrioles are going to have a set of 3 linked together. And how they come together is a little bit different. You've got 9 connected triplets. In terms of our centrioles, and those centrioles are kind of arranged more like a pinwheel than in a circle, okay? And where these are going to become important and where we're going to discuss centrioles again is when we talk about the topic of cell division. So for right now, all I want you to know about centrioles is that they're involved in cell division or mitosis. You can give me either one of those as being correct. I'm not going to get into the detail of what they do specifically in mitosis. I'm going to save that for the video on mitosis because this is a lot to throw at you at once anyway. So those are going to be our microtubules. One of the things that is kind of neat about microtubules are going to be that there are a number of drugs that can interfere with the function of microtubules. And that's going to be really important because some of these drugs, I've got some of these listed in your PowerPoint, but Taxol is a chemotherapeutic drug that's used to treat cancer patients. That Taxol prevents microtubules from forming those alpha-beta dimers. So the microtubules kind of fall apart. How that treats cancer is that cancer are cells that are growing out of control. So they're going through cell division when they're not supposed to. And what that Taxol does is it interferes with the centrioles, it interferes with the microtubules in getting that cell to divide, to make more cells. So if the cell can't make more cells, that's going to be how that particular drug treats cancer. So there are a number of chemotherapeutic drugs that are actually working to inhibit microtubules. On the downside, that's why some of those chemotherapeutic drugs have the side effects that they do, because those basic processes are needed by other cells that are not cancer cells, okay? So I've got a list of them. I've got Taxol in there. Colchicine is another drug that inhibits microtubules. Vinblastine and nicotazole are all drugs that are all target microtubules and different processes associated with microtubules. What I want you to take away, know the names of those 4 drugs and that they essentially inhibit microtubules either from forming or they inhibit microtubules from breaking down. Because what's unique about microtubules that doesn't happen with actin or intermediate filaments is that the way the microtubules kind of move within the cell is that they'll subtract off of one end and they'll add more microtubules onto the other end. So they'll subtract off of one, they'll add off of the other. If you interfere with that process, that microtubule can't move anything within the cell, okay? So that's another way that those drugs can kind of interfere with how microtubules are functioning. If you have any questions, let me know. Thank you for your attention.